Hello and welcome back to G-Fun Facts Online. Today we are going to talk about a shift in human history that is as significant as the Industrial Revolution, but it is happening inside your cells right now. For most of our existence, we have looked at the human body as a machine. It is a useful analogy. We have a heart that acts like a pump, lungs that act like bellows, and nerves that act like wires. But for thousands of years, this machine had a sealed hood. We could watch the gears turn, we could see cells dividing and proteins churning, and occasionally we could throw a wrench into the works, which is effectively what drugs are, to try and jam a broken part or grease a squeaky wheel. But we could not fundamentally rewire the engine. We could not install new software. That era is over. As of early 2026, we have officially entered the age of the programmable human. This is not science fiction anymore. This is synthetic biology. We have moved far beyond the simple cut and paste of early genetic engineering. We are no longer just editing genes. We are installing circuits. Scientists are borrowing the Boolean logic that powers silicon computers, the AND, OR, NOT, and NAND gates you find in your laptop, and they are translating it into the wet, squishy language of DNA and protein. We are transforming human tissue into biological hardware. We are teaching immune cells to run algorithms before they open fire. We are installing patches into cartilage that release drugs in sync with the sun. We are quite literally downloading apps into our organs. To understand how massive this is, we have to look at the hardware of the cell itself. For decades, biology was just about observation. We identified the central dogma, which says DNA makes RNA, and RNA makes protein. We thought of this as a river, a one-way flow of information guided by evolution. But synthetic biology asks a different question. What if we treated that river as a circuit board? In a computer, you have a transistor. It controls the flow of electrons. If the input is high, the current flows, representing a 1. If it is low, it stops, representing a 0. By stacking these transistors, you build logic gates. An AND gate requires 2 inputs to be on for the output to activate. In a cell, the electrons are RNA polymerase molecules, the enzymes that read DNA. The transistors are promoters, which are sequences of DNA that tell the cell when to start reading a gene. By engineering these promoters to respond to specific chemical signals, we can create biological switches. The field had its hello world moment back in 2000 with a genetic toggle switch in E. coli bacteria. It was a simple memory unit consisting of 2 genes that repressed each other. If gene A was on, it shut off gene B, and vice versa. It was BIST-able, meaning it could hold a state, just like a bit of computer memory. But for 20 years, these were just toys for bacteria. They were too slow and too noisy to work in humans. A circuit that worked in a petri dish would fail inside a human T cell because the cell is a cluttered, chaotic environment with thousands of native signaling pathways. But in the last 5 years, and specifically through breakthroughs culminating in 2025, those compatibility issues have been solved. We have moved from simple toggle switches to multi-core processing inside human tissue. Today, the circuits being installed fall into 3 generations, essentially different processor architectures. The first is transcriptional logic. This is the classic approach. You engineer a gene that only turns on if 2 specific proteins bind to its promoter. This is an AND gate. It is versatile, but it is slow. Biology takes time. Making RNA and then protein can take hours. If you need a cell to react to a sudden drop in blood sugar, that lag can be dangerous. That brings us to the second generation, the big leap of 2025, protein-level logic. Instead of waiting for new genes to be expressed, these circuits use proteins that are already made but sitting dormant, waiting for a signal. They rely on phosphorylation, which is the rapid addition of a phosphate group to a protein, to change its state. A landmark study introduced logic gate proteins with smart tails. These are synthetic proteins floating in the cytoplasm. When they detect a specific input, like a drug or a disease marker, they physically change shape in seconds. This allows for real-time processing, which is critical for things like smart insulin. Then you have the third generation, CRISPR processors. If proteins are transistors, CRISPR is the operating system. We usually think of CRISPR as gene scissors, but a version called DCase9 can act as a traffic cop. It can sit on a gene to block it, or recruit activators to turn it on. In late 2025, researchers at ETH Zurich demonstrated a dual-core processor in human cells. They used 2 different CRISPR systems inside a single cell. One core handled metabolic inputs, like sensing nutrients, while the other handled therapeutic outputs, like releasing drugs. This parallel processing prevents the circuit from getting bogged down. So we have the software. How do we install it? You cannot ask a patient to swallow a disk drive. The delivery problem has been the bottleneck of gene therapy for decades. The old way was using viral vectors. We would strip the bad parts out of a virus, like HIV or adeno-associated virus, and stuff the synthetic circuit inside. But viruses are tiny. A complex logic gate requires a lot of DNA code, often too much to fit inside a viral shell. It is like trying to fit the Windows operating system onto a floppy disk. The new standard, which you might recognize from COVID vaccines, is lipid nanoparticles, or LNPs, but the 2026 version is turbocharged. Research in 2025 revealed that standard LNPs were often 50% empty. New manufacturing techniques involving turbulent mixing have created ultra-dense LNPs that can carry massive genetic payloads. These synthetic fat bubbles fuse with the cell membrane and dump the circuit into the cytoplasm. Since they are synthetic, there is no hard limit on the file size. For immune cells, we can even do this outside the body. We harvest T cells, zap them with electricity to open their pores in a process called electroporation, and slide the circuit in. This allows us to rewrite the T cell's genome without using a single virus. Now, let's talk about the killer app for this technology: oncology. The most immediate application is logic-gated CAR T cells. The first generation of CAR T therapy was a dumb missile. It targeted a single antigen. If the cancer had it, it died. But if healthy cells had it, they died too. This toxicity could be lethal. In 2026, we are seeing clinical trials for cells that compute a decision. They use an AND gate. The T cell is programmed to kill only if it detects antigen A and antigen B. For example, a tumor might express both HER2 and mesothelin. Healthy heart tissue has HER2, and healthy lung lining has mesothelin. A standard therapy would attack the heart and lungs. But the logic-gated T cell ignores the heart because it only sees A. It ignores the lungs because it only sees B. It only activates when it sees the specific combination found on the tumor. We also have NOT gates, acting as safety switches. You can program the cell to attack a cancer marker unless it sees a healthy marker, acting as an emergency brake. And for solid tumors, which are notorious for hiding their markers, we have OR gates. If the tumor stops making protein X to hide, the T cell is programmed to recognize protein X OR protein Y, making it impossible for the cancer to escape. But while cancer gets the headlines, the application for chronic disease is perhaps even more profound. This is where we move from killing bad cells to regulating good ones. Take rheumatoid arthritis. It follows a daily clock, with inflammation spiking in the early morning. Taking a pill when you wake up is often too late. In early 2025, researchers unveiled a chronogenetic circuit. They engineered cartilage cells with a circadian clock. They linked a therapeutic gene to the body's natural rhythm. These cells wake up in the pre-dawn hours and release a pulse of anti-inflammatory medication before the patient even wakes up. This is temporal targeting. We are also seeing the next version of the artificial pancreas. Smart beta cells are being engineered with a closed-loop logic gate. If glucose is above 150, they produce insulin. If it drops below 70, they secrete glucagon to raise it. Because they use that fast protein-level logic I mentioned earlier, they react instantly, mimicking a healthy organ. Of course, you might be asking, if this is so powerful, why are we not all cyborgs yet? There are bugs in the system. The first is cellular burden, or the RAM limit. A cell has finite energy. When we force it to run a complex dual-core processor, we steal its resources. This is called metabolic burden. The cell grows slowly or dies. Even worse, evolution hates these engineered cells. Over time, they mutate to break the circuit and save energy. To fix this, modeling papers in 2025 proposed Punisher circuits. These are genetic kill switches linked to the circuit's function. If the cell tries to delete the therapeutic app, the Punisher activates a toxin and kills the cell. Only the obedient cells survive. Then there is the blue screen of death, or immunogenicity. The body is paranoid. It scans for non-self proteins. Many circuits use parts from bacteria. If the immune system spots them, it destroys the therapeutic cells. The field is now moving toward humanized parts, building switches entirely from human protein sequences to make the circuit invisible. There is also the problem of retroactivity, or signal noise. In biology, measuring a molecule usually means binding to it, which consumes it. Your sensor can accidentally drain the signal it is trying to detect. Engineers are currently designing insulated buffers to prevent this. Looking forward to 2030, the field is getting truly sci-fi. We are looking at organoid intelligence. Silicon chips are hitting physical limits and overheating. The human brain runs on the energy of a dim light bulb. Researchers are growing brain organoids, tiny spheres of human neurons, and wiring them into biocomputers. These living processors can already play simple games and perform speech recognition. We are also seeing the rise of the Internet of Body. Cybergenetics is the interface between digital and biological. We have circuits that respond to light or magnetic fields. Imagine a smartwatch detecting a fever and sending a digital signal to an implant in your arm, which triggers a red LED, activating a light-sensitive gene circuit to release ibuprofen directly into your bloodstream. But with this power comes heavy ethical responsibility. There is the dual-use dilemma. A circuit that detects a tumor and releases a cure is a miracle. A circuit that detects a specific genetic ancestry marker and releases a toxin is a bioweapon. The same chassis can carry a good or bad payload. We also have to redefine what it means to be human. If you replace half your liver with synthetic computing cells that are smarter than the original organ, are you still fully human? And finally, the kill switch debate. Current ethics guidelines suggest that all high-level synthetic circuits in humans must have a remote-controlled emergency brake, a drug-inducible suicide gene, in case the therapy goes wrong. We are transitioning from the user of the human body to the administrator. The cellular logic gate represents the convergence of biology and engineering, turning the chaos of life into a solvable problem. The operating system is live. The hardware is ready. And the update is downloading. Thanks for listening to G Fun Facts Online.